What is nutrigenomics? Nutrigenomics is a study of the effects of food and food constituents on gene expression and how genetic variations affect the nutritional environment. Food affects living organisms differently. Therefore, nutrigenomics studies the effects of food on the genetic and epigenetic level. The recent development in nutrigenomics research can help us improve aquaculture nutrition. It can help us understand the molecular mechanisms of nutrition in aquaculture organisms, and thus can help us improve nutritional performance of aquaculture organisms in a more comprehensive way. So how does nutrigenomics can help us improve aquaculture nutrition? Nutrigenomics is useful for evaluating the response of organisms to the nutrients, identify the factors responsible for metabolism, diet development by optimizing the dietary nutrient requirement of a particular species. It can also facilitate through understanding nutrient responses in cells and complementing basic husbandry practices. Other than that, nutrigenomics can also facilitate in knowing organs and tissue specificity in utilizing different nutrients. We can also understand basic metabolic changes by dietary factors and many more. Nutrigenomics research in aquaculture nutrition is a relatively new field of study. In Malaysia specifically, nutrigenomics application in aquaculture nutrition is still fairly underdeveloped. However, we think that nutrigenomics research in aquaculture is a field that is very important and beneficial to study, and its application can help in the development of the aquaculture industry.
Nutrigenomic impacts on aquaculture. Minimize fish oil use in feed. Fish oil is obtained from wild fish and it's an important component in fish feed because it provides essential fatty acids such as DHA and EPA. However, due to its limited source, fish oil is quite expensive. Therefore, by using molecular techniques on fin fish to alter their lipid metabolism, such as reducing their lipid need and optimizing their lipid metabolism, the use of fish oil can be reduced in developing new fish feed and hence reducing the overall cost. Enhancing fish immunity. Nutrigenomics mainly have been applied to enhance fish immunity through nutritional supplements and feeds. The feeds will target specific genes that can induce immunity and also increase fish growth rate and performance. Target specific genes to increase fish performance. Through the applications of fish feed and nutritional supplements, genes related to environmental stress and stress tolerance can be induced to increase fish performance and quality. Faster growth rate and better quality meat can be achieved. Limitation and key areas for improvement in nutrigenomics. Lack of data or genomic information on aquatic species. Aquatic ecosystem is a diverse ecosystem filled with thousands of different species. Therefore, data and genomic information must be obtained before culturing a certain type of fish and applying nutrigenomic applications onto the aquaculture system. Same species organisms may also evolve when exposed to different environmental conditions, which means they are no longer genetically similar and more studies must be done to collect the genomic data of the species. Effect of feeds towards fish physiology. Due to diverse genomic of aquatic species, the effects of feeds towards fish physiology may vary, which means some fish may benefit and flourish from the feeds and some might die. Therefore, individual and specific studies must be done on cultured animals to meet each one of their needs by developing specific feeds.
Future study. Fish lipid nutrigenomics. Lipid is an essential nutrition for fish, which it will give them the energy for growth, development, and reproduction. Fish oil will be used for feed production in order for the lipid to be supplied to the fish. In order to gain fish oil for aquaculture, wild fish were caught in order to produce fish oil. Since the aquaculture industry is expanding, the demand for fish oil is increased. Hence, many wild fish were caught, and this can affect the wild fish population in the nature. By using nutrigenomic approach, we can reduce our dependency of wild fish to supply fish oil to our aquaculture fish. This can be achieved if we have a better understanding of lipid storage in fish and muscle growth. Next is nutrigenomic and immune function. Fish are exposed to million microbes in the sea, which means they are exposed to diseases that can increase the fertility rate of the fish, and hence farmer will experience losses. By using nutrigenomic approach, normal feed, which is usually being used for nutrition purposes, can be upgraded to feed nutrigenomic, in which it will help to increase the immune system of the fish. In order for the nutrigenomic feed to be effective in improving the fish immune system, we can achieve this by using omic technology, in which there is involvement of the transcriptomic and proteomic. Next, by using molecular pathway enrichment analysis, we can study how certain feed can alter certain genes, especially genes that involve in immune system of a fish.